Alrighty, good morning. Going to, we're going to go over, we're going to learn about the microsporogenesis, megasporogenesis, and embryogenesis and angiosperms. So basically, we're going to kind of learn about the life cycle of some angiosperms. I know we've talked a little bit about mega and microsperms, so I know you have a brief idea as to what those are about. So first thing I want to go through is this idea of alternation of generations. Every organism kind of goes through these stages, but not everyone considers them a stage in their life cycle. Unlike the plants actually consider this a stage. We consider this more of like a process because we don't consider a stage of our development as formation of gametes. It's just a process that we go through. So the two different generations that your plants are going to go through are going to be the sporophyte and then the gametophyte. So the sporophyte is going to be able to produce your spores. And at this particular stage of development, or this particular life cycle stage, the plant is considered to be diploid. So it's got both sets of chromosomes in every cell, or both copies of each gene. That spore will then go through meiosis, and you will get to the gametophyte stage of development. This is where you produce the gamete, which will be haploid, meaning it's only got one copy of those genes. And then it will go through mitosis to restore that number of chromosomes. So first I'm going to go over these parts, and then we'll hit the diagrams. So sporogenesis and gametophytes. Sporogenesis is the process of making your microgametophyte, which in this case is the male gamete. Should be the sperm. I'm not sure why I put male gametophyte. I mean, it's technically the male gametophyte. So, but a more common word might be the male gamete. Then your mature microgametophyte is going to be the pollen grain. There will be a stage where you will have an immature microgametophyte. So we'll talk about that here in a minute. And then we've got your megasporogenesis, which is the process of making the megagametophyte. Or in this case, it's the male gamete, which is the egg. Don't confuse that with the ovule. The ovule contains the egg eventually. Or will give rise to the egg. The mature megagametophyte is going to be the embryo sac. And then I'm going to give you these two things. They're not going to make a lot of sense until after we go over the diagrams, but I'll give them to you anyway. First part is the ovule. The definition of the ovule is it's an integumented megasporangium that surrounds or encloses the nucleus. Sorry, not the nucleus, the megaspore mother cell. We haven't talked about the megaspore mother cell yet, so don't worry. The second one is that eventually your ovules will give rise to seeds. These little things called integuments will give rise to the protective outer coat of your seed called the testa. And this is necessary for when you have your embryo, your little baby plant. So now we're going to look at your diagram. So go ahead and take a look at those diagrams for me. So the very first set of pictures is the stamen that I've drawn for you, the anther and the filament. I then took the anther and did a cross section through it. Okay. In each of the corners, there's this mass of little cells. That cluster of cells is called a microsporangium. Inside of the microsporangium, you've got these small microspore mother cells. These are diploid. This is the microspore site, sometimes they're called. That's what I've seen some of them called that. So now you're going to take that microspore mother cell. We're going to make it bigger. The next picture. I didn't label it. I just put it as a diploid, the 2N. That microspore mother cell, that's your spore, by the way. You're then going to go through meiosis and become the microspore, the haploid microspore. Specifically, you're going to get 4 because you know that meiosis yields 4 haploid cells. So there they are. We're going to take one of those microspores and it's going to go through mitosis. It doesn't actually separate, but it does make 2 nuclei. So the microspore has 2 nuclei. One of them is called the generative nucleus. The other one's going to be called the tube nucleus. At this point, you have an immature gametophyte. So the generative nucleus needs to go through another round of mitosis. The tube nucleus is kind of a vegetative cell. It doesn't do much after this. So that generative nucleus undergoes mitosis again, and you end up with 2 sperm nuclei or 2 sperm cells. There's a wall called the sporopollenin that kind of goes around this whole microspore. And this is now, because once it has that, the wall of the sporopollenin and the 2 sperm with the tube nucleus, you now have a mature male gametophyte. Or in other words, you have a pollen grain. So then they're just going to hang out there until pollination, waiting for something to pollinate them. Okay, down below, this would be the megasporogenesis. So you've got your ovary with the 3 parts there. Specifically, we're going to look at the ovule, which is held on to by the funiculus. That holds on to, that anchors the ovule to the inner lining of the ovary. So then I've taken that ovule and I've kind of blown it up. So you can see the funiculus. And now we've got the, it's not technically an ovule yet, but you've got this megasporangium structure inside of the not quite ovule, and that is diploid. You can kind of see the little appendages there on the bottom. Those are called the integuments. So now that should kind of make sense with the that second or the third bullet under the megasporogenesis. The ovule gives rise to seeds. The integuments right there, those are going to give rise to the protective outer seed coat later. They are a diploid also. That there's an opening there in between the 2 integuments. It's called the micropyle. That'll be important later because that's where the pollen tube will attach to. Now you've got your megasporangium there, inside your megasporangium, just like the microsporangium, you're going to have a megaspore mother cell. This is also diploid. Sometimes you'll hear this one called the megasporocyte. So the megaspore mother cell will undergo meiosis. Just like with the microsporogenesis, it's going to yield 4 megaspores. However, this one's a little different. 3 of the megaspores will abort. So you'll be left with only 1 surviving megaspore. Still diploid or still haploid, but only 1 megaspore survives. That megaspore will then go through mitosis. I've kind of pulled it out in the next picture until it's got 2 nuclei. It's going to then go through another round of mitosis till it gets 4. And then it's going to go through 1 more round of mitosis until it has 8 nuclei. That's where you get to that funny looking picture with all 8 nuclei. You have 8 nuclei, but you only have 7 cells. You've got 3 of them there at the top that are opposite of your micropyle. Those are the antipodal cells. These are going to be important later because they're going to provide nutrition for the embryo. That middle set is the polar nuclei. It's not technically a diploid because those nuclei haven't fused. So it's an N plus an N. But this will be important later. We'll do something with this polar nuclei. The fusion nuclei is what I saw it some people referencing it as, but I was taught polar nuclei. Now the bottom 3, the 2 outside cells are called the synergid cells. The 1 cell in the middle is the egg cell. This whole structure is considered the mature gametophyte. Okay, I lied, sorry. The ovule, it's still an ovule back at the beginning because it's got the integuments and it's got the megasporangium, so I apologize. That structure is the ovule, sorry. Doesn't have the egg yet, but it will eventually. So now those 2 sentences should make sense. The ovule is the tegumented megasporangium, which it is, little integuments around it, and it's surrounding that megaspore mother cell. Makes sense. Because it does. So hopefully that makes some sense now. Okay. Sorry, I'm making sure I've got everything. All right, if you flip your page, not to the doodles, but to the next set of doodles that is not in my handwriting. At the bottom, it says page 54. You're going to see the general alternation of plants part. So if we kind of look at this in plants, and this is very similar to humans, we'll just start with the sporophyte. You end up with your sporophyte, which is diploid, goes through meiosis in the sporangium to create your spores, which then go through mitosis. And then you get your gametophyte parts, which will lead to and you're only going through the mitosis to get your nuclei. Now humans don't go through that part. We skip kind of all together. We just go from that quote unquote sporophyte of the adult stage I guess it's not technically an adult for females, but you go through that diploid stage and then you create your gametes from there. And then you get your sperm and your egg cells. They're going to go through fertilization, and then you get a zygote, which goes through mitosis, and you get your big part of the plant. Now, that might be like a, what is that? Might be like a bryophyte. I think I saw. But then with the alternation of generations, there are two distinct, sorry, that's our dog drinking. You've got your spore, we'll start at the top again with your sporophyte. You have a mega and a microsporangium in your ovary and in your anther. It's going to give rise to your mega or microspore mother cells, which go through meiosis to make your mega or microspores, which then go through mitosis to gain more nuclei, and you end up with your megagametophyte and your microgametophyte, which would be your ovule, your pollen grain, your embryo sac, and your pollen grains. And inside of those, they contain those gametes, the egg and the sperm. Okay, so that's the one generation. You then get your fertilization to occur. You get your zygote, go through mitosis to get an embryo, go through more mitosis to end up back at your sporophyte. So there's two distinct stages there, as opposed to a part. So, okay. Okay, so now go to, we'll move on to this next one, this next slide. Okay, we'll talk about it again, just like with the last page. We'll talk about this first because we'll get you the information, then we'll go through the diagrams, and hopefully it'll make more sense. Angiosperms are the only group to go through this thing called double fertilization. Okay, basically what's going to happen is in fertilization is there's going to be some enzymes that are going to digest through that wall of that ovule to get to the egg cell. They're going to do that through the micropyle. They're going to make this long tube called the pollen tube, because the pollen is normally stationed out on the stigma. So they're normally out on the stigma. So they create that pollen tube to go down through the micropyle. Just like with human reproduction, only one sperm fertilizes the egg to get to the zygote, to make the zygote. Now here's where double fertilization comes into play. I told you we needed to talk about the polar nuclei in that second sperm. The second sperm will fertilize that polar nuclei, and you will end up getting a 3N nucleus, or 3N structure. So it's got a triploid cell, and we're going to call that an endosperm. Okay, so look back at your diagrams for me. So I've drawn you, so I've drawn you the picture of the ovary there. The pollen is up on the stigma, creates that pollen tube. It's going to go down through and connect to the bottom of the micropyle. And then that's where those sperm, and I've drawn that to the right. You got your gametophyte, the pollen tube, and you got your tube nucleus that's just hanging out, doesn't fuse with anything. Because remember, that's that vegetative cell. And then you've got your two sperm that are going to fertilize. So flip to The next doodle that I did not draw, so it's on the back side of the alternation of generations photo. So it's got a diagram of the flower itself there. So remember, we're in Angiosperms. And so it's showing you how the anther makes that microspore mother cell, how the ovule makes the megaspore mother cell. Again, how they go through meiosis to make your mega or microspores. The 3 surviving megaspores, the 3 of the megaspores abort, 1 survives. It's just taking one of those microspores, putting them through mitosis. You end up with the generative nucleus of the tube nucleus. And you've got your 1 surviving megaspore. Both of these go through mitosis, or microgametophyte ends up with those 2 nuclei, with the 2 sperm and the tube nucleus. And then your embryo ends up with the 3 antipodal cells, the 2 synergid cells, the polar nuclei, and the egg cell. Okay. Now, if you follow the arrows, one of the sperm will fertilize the egg to make that zygote. The other sperm is going to fuse with that polar nuclei to make that triple fusion endosperm, which will be needed later for the embryo to develop. And then that zygote goes through, becomes the embryo, which will eventually become the mature sporophyte, and then we'll start all over again. But that kind of shows you the 2 generations, because that line is there. But that's what we mean by double fertilization. If you flip to the next page of drawing not drawn by me, it's page 56. This one kind of shows you a bigger, better drawing of your mega and micro gametophytes. So now I've been using the term the antipodal cells. They're not really cells for all intents and purposes. They're just a set of chromosomes in a nucleus, specifically haploid. So, but that kind of shows you a little bit more on the mega and microgametophytes. Okay. So the next part, embryogenesis, making of the process of making the embryo. Just like in humans, after fertilization, you have a zygote. That zygote's going to go through mitosis, and it's going to turn into a basal cell and some suspensor, a suspensor. Good morning, Mommy. Good morning, buddy. The basal cell is upstairs, bud. Oh, upstairs? Upstairs. Okay. Sorry about that. So you have your large basal cell. And if you look at your drawings that I drew you, I know you're flipping back and forth, you can see that basal cell at the bottom, which is the fertilized zygote, or not quite an embryo yet, it's fertilized zygote. So it's going to start going through mitosis to make the suspensor cells. So it's just like the placenta. It's kind of like the placenta. It's got to anchor into the parent to get that nutrition. Because remember, this is the ovule that's attached to the inside of the ovary. So it's still connected to the parent plant. So then the suspensor will then grow to form the rest of the new plant. Your embryo then will go through 3 different stages: your globular stage, your heart-shaped stage, and your torpedo stage. So it's just a matter of what they look like. It has nothing to do with anything other than just what they look like. Your globular stage, it literally just looks like a circle or a globe. So the heart-shaped stage kind of looks like a heart, and then the torpedo stage is once it starts growing kind of long. Appendages, I might call them, they're like appendages. So, if you look, I don't know if it's the next one or not. I don't know if I'm getting ahead of myself. All right, so I'm going to describe the last doodle that I drew. So now it looks kind of different. You've got your testa from those integuments. And that's that hard outer coating there, and that's going to be your seed. You still have that basal cell. You still technically have your suspensor, but now your suspensor has developed into other parts, including your root apical meristem, the beginnings of your root apical meristem. And then you've got technically your embryo, which is diploid now. And then you've at the very other end there, you've got your shoot apical meristem. And then past that, you've got your cotyledons. In this case, there are 2 cotyledons, so this particular doodle that I drew you would be a dicot. But this would be considered a mature embryo at this point, or basically a seed. If you look at the last page of doodles that I did not draw you, it's page 57. You can kind of see the embryo there. It's going to go through the embryogenesis here. So the pollen is up at the top. It's developed that pollen tube. The other pollen is building its tube, but the other one already is connected with another ovule. So then in one, you can see that the pollen tube, it's got the 3 nuclei. Your 2 genitive and your 1, well, your 2 sperm, technically, and your 1 tube nucleus. In picture 2, the pollen tube has infiltrated and it's getting ready to, the sperm are getting ready to go into the megagametophyte. Picture number 3, it's released the 2 sperm. Picture number 4, you've got 2 big structures there in the middle. The one in the center is the tube nucleus because it's got the 3 nuclei, and then the other is this fertilized zygote. 5, the synergid and antipodals are becoming what they are going to become, the nutrition. You can kind of see the integuments are starting to fuse to start making that outer covering. The egg is going to anchor in picture number 6 to the inside of the ovary. And it's going to start that suspensor. Picture number 5, we are now in the globe stage, the globular stage. Picture number 8, we're kind of in the heart-shaped stage, sorry. And then when it starts to fold over on itself, it would be the torpedo stage. This one is also a dicot. If it only had one cotyledon, it would be a monocot. So that's essentially embryogenesis for you. I know it's kind of crazy with all the doodles. I mean, we thought like photosynthesis was a lot, but kind of puts photosynthesis to shame. But if you do have any questions, please don't hesitate to ask. Like I said, I know it's getting to be a little more confusing. So if you do have questions, just let me know, and I'd be more than happy to answer. So, oh, I forgot the seeds part here. I was getting ready to wrap up, and I didn't have this part. The last thing, all seeds, which we'll talk about here in 2-3 days on Thursday. All seeds are going to have the embryo, 1 or 2 cotyledons, and then your shoot and root apical meristems, which makes your mature sporophyte. And then it's got that testa, that tough outer protective coat, and stored food from your cotyledons or the endosperm. So, all right, so pretty much wraps it up. Like I said, if you do have questions, please don't hesitate to ask. Otherwise, have a great day.